Okay. So I believe everything is fine now. So sound is also fine. So this lecture is now currently being live streamed on YouTube. And I'll be checking also the YouTube chat occasionally. And if you have any questions about the course or anything in general, you can write in the chat. So with that, we can start the first lecture, I believe. So welcome everyone to the first lecture of PNS Genomics. Our title for this week is the PNS course introduction and the scope. And before I get into basically the details of this lecture, I want to briefly introduce myself. I'm Can. I'm a PhD candidate and also a lecturer at Safari Research Group, led by Professor Onur Mutlu. My research interest actually is in the intersection of bioinformatics and computer architecture, which includes real-time genome analysis, similar to search in a large space of genomic data, hardware algorithm co-design to accelerate the genome analysis, and also genome editing and error correction. So you can get to know us and also our group, our research, visiting our website. And also you can contact me using my email address. Also, you can learn more about my research that I do in our group in Safari using this particular website. And also you can follow my academic tweets on Twitter or also known as X nowadays. So let's see what we have for the agenda for today. So we're going to be briefly covering the goal of this PNS course. And also we're going to cover what we expect also from students for a successful execution of the course for both of us, including the mentors and also the students. And also we'll have some brief introduction to genome analysis today. So let's cover the key goal of this course. So one key goal of this course is to understand the computational steps and challenges in genome analysis. So we want to enable students to understand these challenges. And also the second goal is to learn how to overcome these challenges with efficient software or hardware-software co-design so that we can achieve fast and accurate genome analysis by overcoming these challenges. So to achieve these goals, we offer two Genomics, PNS Genomics courses this semester. One is called PNS Mobile Genomics. The other one is called PNS Accelerating Genomics. Although in low level, the definitions are slightly different, in high level, the challenges we're tackling are essentially identical. So both of these projects are project-based courses. Both of these courses are project-based courses, which essentially will aims and also hopefully will enable understanding and improving the cutting-edge techniques by doing some hands-on research exploration with mentors. So you're going to be assigned, students are going to be assigned some projects so that they can work with their mentors. And also we have optional weekly lectures on genomics, including this one, so that you can essentially also acquire the technical skills and also the background on genomics, which is usually handy to have fast and effective progress on the projects that the students are going to be working on. And also by hopefully completing these projects successfully, you're going to be improving your research and presentation skills. So there are two aspects of these courses. One, as I said, the hands-on projects that the students are going to be doing with their mentors. So what we're essentially going to be doing is the students are going to be experimentally evaluating the heuristic algorithms and also the architecture to observe their effects on the end results. Based on the project that you're going to be assigned on, in high level, that's what the students are going to be exploring in general. We also have, as I said, optional week lectures. These lectures will cover the basics of genome analysis to understand the computational steps of the entire genome analysis pipeline. Although following these lectures are not required and they are optional, we strongly suggest students and everyone to follow these lectures so that to achieve essentially two main goals. One is to get the basic knowledge in genomics, and the second is to also understand the cutting edge research essentially in genome analysis. There are many key benefits of this course. We essentially, as mentors, aim to improve following aspects of students. For example, students can improve their basic knowledge in genome analysis. Also, they can improve their technical skills in genome analysis by doing a project with us related to this direction. And also, they can improve their critical thinking and analysis. Also, students get familiarity with key research directions. And at the end of the semester, students will present their projects and their also progress. And this also hopefully improves the technical presentation skills of the students, essentially. So there are certain prerequisites of the course. The first item that I want to mention in this direction is we don't really require any prior knowledge in bioinformatics or genome analysis. So you don't really have to have this knowledge to make progress on your projects. Your mentors will be giving you the basic knowledge whenever necessary. And also your mentors may also point you to certain lectures so that you can watch these lectures and also get this necessary background knowledge to complete your project. But what we essentially require is an interest in optimizing efficiency and solving complex problems, of course. You need to be motivated, essentially, to complete this course. We also require a basic too good knowledge in C or C++ programming language. So if you don't have any background on C or C++, depending on the project, essentially, there may be some projects that really may not require C or C++, but in most cases, based on the previous semesters, in most cases, we really require these programming languages. So if you don't have this knowledge, there's a high chance that you're going to struggle a lot and you're going to spend most of your time essentially understanding the code or essentially learning how to code in C or C++. So that's why we really essentially ask you to check essentially if you have this knowledge in C or C++ so that you can have a smooth progress this semester. So other thing that I want to mention is you really need to also have some familiarity with accessing servers. For example, using SSH and also proficiency in using the Linux terminal as well. So this is really required. So if you don't have this knowledge, we actually strongly suggest you to drop this course because this is almost the first, the tasks essentially that are assigned to you from the first week will essentially require you to access our servers, which will provide the details on how to access the servers and how to use the server. But essentially the basics, for example, using SSH and also being comfortable with using the Linux terminal is really required so that you don't waste your time learning these where you could essentially invest your time in improving these, in basically making progress in your project. So that's why you should really have some familiarity and also proficiency in using Linux terminal. Also, if you have taken some related previous coursework, such as digital design and computer architecture or equivalent course, this is really desirable, but again, not required. And also, depending on the project that you may be assigned to, we seek some experience in either FPGA implementation, or GPU programming, or algorithm design. If you have essential background in any of these, it's really beneficial, again, depending on the project that you may be assigned. So if you, for example, don't have any background on GPU programming, you don't have to worry about it, because then this means that you're not going to pick a GPU related, let's say, a project, but rather something else that fits your, let's say, background and your, let's say, motivation. So let's cover some then course requirements and then also the expectations from the students, as well as the prerequisites that we just covered. Again, weekly lectures are optional, but strongly suggested to follow. So these lectures are going to be on our course website. So you should already have the link to the course websites from the course catalog at ETH, or essentially from YouTube. You can also see the playlist of this course and then see basically what's next. So talking about YouTube, we're going to be usually either live streaming or premiering the lectures on YouTube. There is no in-person meeting for lectures. There is no Zoom link to join. What you just need to do is just find the YouTube link, either on YouTube using by either checking Professor Mutlu's YouTube channel, or more easily going to the course website and then seeing the links that we're going to be including in the course website. So that's essentially about the weekly lectures. Projects, which are the core part of this course, what you're going to be doing is you're going to be building, implementing, and designing under close mentor guidance. We expect that you're going to spend around 6 hours of work per week for the projects that you're going to be assigned to. And also we expect you to hold regular meetings, for example, weekly meetings with your mentors, so that you can describe your progress, and so that the mentors also can give you the right feedback, so that you can continuously can do the right progress that you should be doing until the end of the semester. So these projects are going to be assigned to you after the mentors present their projects to you. So we're going to hold a session for it, which will be announced internally. We will email you, and also we will post post a message on Moodle, which you will also receive via your email, where you're going to also see the details about where this project presentations or project introductions are going to be held. This will be both in person and also over Zoom. So you have the option to join either in person or using Zoom. And here you will essentially listen to the projects and then see the details of and expectations of the projects. And then later on, we're going to ask you to rank some of the projects that you think that you're interested in. And based on your ranking, we're going to be assigning you the projects, and the mentors will contact you accordingly about, and will describe you the further steps about this project. And by the end of the course, you're going to be presenting your progress to your peers and also to the mentors, which will hopefully will be enough for you to complete this course successfully. So what is really important is essentially the communication with your mentors. You should regularly check the course website and also the Moodle, Discord, and the emails. Our instant communication channel is usually Discord, but you're free to choose anything with your mentor, so you just need to discuss it with your mentor. Also, we expect you to have some timely responses to mentors. As I mentioned, at the end of the semester, you're going to be presenting the project that you worked on, and also you're going to be presenting your progress, the results, for example, that you gathered. And this will happen in the last week of May at the end of the semester. So you're going to be also submitting your materials, for example, the code, if any, the results that you're going to be uploading to them to some repository. We will provide all these details essentially later on throughout the semester. And essentially, if you have some significant progress in your project, or if you want to continue working on the project, we can support you for publication later on. So you can just let us know that you want to further pursue this project for publication, and your mentors will be more than happy to help you for this purpose. So a bit more detail about project assignments and progress. So as I said, we assign the projects based on your interest and skills. After mentors present their projects before the assignment, and then you rank these projects based on your interest, and then mentors will be contacting you to arrange regular meetings. Again, to repeat, regular meetings are critical to show your progress. So with that, let's then cover who your mentors may be. I'll start with myself. Again, I'm John. I'm one of the mentors and also the lead lecturer of this course. We also have postdocs, PhD students, master students in our group who can be your mentors. We have Hayu, a postdoc and a senior researcher in our group. We have Nika, who is a PhD student. Also, we have Joel, who is again a PhD student in our group. We have Banu, Arvid, master students, and also Zulal, a visiting PhD student in our group. So these people will present some projects to you, and then you'll essentially pick or rank these projects based on your interest. Our weekly lectures are on the course website, and you can already see the tentative schedule on our course website. So we already have the link to this YouTube live stream again on the course website. So you can find all this information, including the links to the slides, to these slides on the course website. So it's pretty useful. Please, if you want to follow the lectures, go and check the course website and then see what's in there. We've been giving these lectures, the PNS Genomics lectures, for many years. I believe it's been 3.5 years so far, and we've been live streaming again these lectures. So you can find the similar content again from last semester already online on YouTube in this particular playlist. So you can first find the links to these slides on the course website, and then you can go and find the links to these essentially YouTube links, and also as well as other papers and the other links that I'm going to be showing you in the coming slides. So if you already want to essentially watch some lectures, you can already go and see what's there in the playlist from the previous semester. Again, these are the links to even older semesters. So with that, we've already done with some basic introduction, let's say, to the course, the goal and the requirements of this course and the expectations and so on. So let's then briefly cover some basic introduction to genome analysis, although we're going to be covering more, let's say, about genome analysis, about the steps in genome analysis in the coming lectures. But essentially, I want to really give you some gist of it. What is really, why this is really important, and what we're really doing essentially in genome analysis. To motivate you, we really essentially need faster, scalable, and accurate genome analysis, because genome analysis is important for many reasons. For example, it is important for understanding the genetic variations, species evaluation, and so on. It is also very important for predicting the presence of relative abundance of, for example, microbes in a sample. So we may want to understand what is the percentage of a particular pathogen, for example, in the environment that we're living in, so that we can take some early actions. And one example to taking some early actions is, of course, the rapid surveillance of disease outbreaks. Right? We may want to quickly understand whether a particular pathogen is being developed in a particular community, and before it gets really threatening, maybe we want to understand what are the actions that we can take so that this pathogen doesn't essentially become essentially threatening to that particular community. And also, of course, genome analysis is really, really important for developing personalized medicine. By understanding each individual's genome, we may, for example, be able to instruct or essentially come up with, let's say, particular drugs that are really personalized for that individual. And if you look at how we perform genome analysis in real life, it really starts with sample collection. So sample can be your blood, it can be coming from your tissue, or like any cell, for example. It may be coming from an animal, plant, or anything, but any living cell that contains a DNA or RNA, let's say, is collected somehow. And this sample is then goes into a certain procedure that we call library preparation, so that, for example, the DNA that we want to analyze is amplified. It's duplicated, let's say, in the wet solution that we have, so that we analyze it, let's say, in a higher resolution, and some other, let's say, procedures to purify and so on. And so what then happens is that the genome, the entire genome we have, for example, in human genome, we have 3 billion characters in one strand of our DNA. That gets chopped, essentially. That gets fragmented into small pieces because of certain limitations, let's say, of sequencing technologies, which will also cover why these DNAs are chopped before essentially producing the output. And the output from the sequencing machines, so these machines are really responsible for essentially translating the biological information into digital data, let's say, into a human readable form. So what they do is that they get the DNA, they get this library prepared earlier as an input, and then they output the digital data, which is the essentially the representation of the DNA of the genome that we want to analyze. And this is what we are really, these two steps are what we're really interested in, essentially, because we really want to analyze the digital data to generate some insight about the genome of a particular individual, let's say. And as I said, one key step is to input the biological information into the sequencing technologies so that they can output us the digital data. And there are many sequencing technologies nowadays. For example, Oxford Nanopore technologies, PacBio, Illumina, and they each have certain limitations, certain benefits, certain, let's say, conditions, let's say, to produce the output. We're going to be covering these as well, I believe, next week in our lecture. And we continue to look at these newer genome sequencing technologies. For example, we even have these handheld sequencing devices that are literally, you can literally put them in your palm, and then these machines can essentially generate the genomic data that you may want to analyze, even in real time, let's say, so that you can also perform your analysis in real time. So we look at these newer genome sequencing technologies, understand their limitations, bottlenecks, and also their opportunities continuously. And if you are interested in learning more about, for example, one of the newer sequencing technology known as Oxford Nanopore technologies, you can take a look at this paper. But what we really see is that there is a bright future for analyzing the genomes intelligently, because there are certain conditions, certain limitations that each, let's say, genome analysis application may require. And when we talk about intelligent genome analysis, we really think about many, let's say, aspects of the analysis. For example, we want fast genome analysis. Of course, this comes with certain latency and throughput requirements. We want to make large-scale analysis. For example, we don't really want our algorithm to be applicable for a very small, for example, a viral genome, but it really also needs to be applicable to an entire population of, let's say, the human samples. And of course, we want to make an accurate analysis, right? If the analysis that we're making is inaccurate, it really isn't important whether it is extremely fast, extremely efficient, because the analysis that we make out of it is really, really important, is really critical, is really sensitive analysis. So we want to make it very accurate analysis. And we want to use intelligent architectures. Again, this buzzword, this keyword comes up again for this particular item, which I'll show you in the next slide what this may mean. But essentially, the goal is to enable energy efficient and high bandwidth, let's say, the analysis. And of course, DNA is extremely sensitive, extremely valuable asset. This means that the privacy is very important for this type of analysis. And you may also have heard of this 23andMe breach that recently happened, where genomic data of maybe thousands or maybe millions of people was leaked, essentially, and which is very dangerous, let's say. We will try to motivate why this is dangerous in the coming weeks. But if you're interested in learning more about what an intelligent genome analysis can be and what are the overview of steps that we take in genome analysis, you can go and check this paper. But what we continue to see is that the problem with the genome analysis today is, although we use these sequencing machines that are really, really fast for data generation, because this is why they are what they are specialized at, right? So they are specialized at generating the data at a high throughput, not really at analyzing this data, let's say, at high throughput. So although we can generate this data at a very fast speed, the machines, the computing systems that we use to analyze this data are essentially slow. And the main reason for that, they are not particularly designed to analyze this specific type of data, right? That has specific requirements, that have specific profiling and so on. And that's why they incur many, let's say, bottleneck, many limitations. And that's why the genome analysis today is not really at its best. Let's say, let me put it that way. And to make it faster, to make it efficient, to make it even maybe more accurate, we look at intelligent architectures. For example, modern systems. We have really, let's say, the heterogeneous environment of computing systems today. We have FPGAs, we have GPUs. We can even use, let's say, processing. We can even use memory technologies today to compute data. Even we can perform computation within the flash, within the SSD, depending on the needs of the essential application. We essentially look at all these architectures and ask ourselves the question where this particular data needs to be analyzed, let's say. And of course, there are even, let's say, particular conditions or requirements for certain applications in genome analysis. For example, there are certain devices that can be, there are certain sequencing devices that can be plugged into mobile devices, right? Maybe your mobile phone, maybe your laptop, let's say. You may connect the sequencer to this mobile device, and then you may want to analyze the genome on the go, right, in a portable way. So in this case, really the energy, the power usage, let's say, the power requirements, and of course, the speed of the analysis is really important there. So depending on basically the platform that you're going to be doing your analysis, the requirements or essentially the architectures that you may want to use, even the algorithm itself that you may want to use may change. So we keep all these aspects in mind to achieve intelligent genome analysis. And to achieve this, we essentially look at the entire stack of computer architecture, almost even starting with the problem itself, problem of genome analysis itself, down into the device level, into very low levels, let's say. We ask ourselves, where is the problem, right? So what can we change in any of these steps so that our analysis becomes more efficient, right? It becomes faster, energy efficient, and even more accurate. So we really look at the entire stack of computer architecture to achieve this. And in the next lectures, we're going to be covering many aspects, many steps in genome analysis, starting with essentially how these sequencing devices work, what is the type of data that they get as input, and what is the output from them, and what are their limitations, what are the opportunities from these sequencing technologies. And then next, we look at how we analyze this digital data that are generated from these sequencing technologies, how they are essentially translated into, let's say, meaningful information by covering certain steps in genome analysis. And we're going to be covering all of these in several weeks. So again, I want to show you the tentative schedule of our course. We're going to be starting with the fundamental steps in genome analysis, and then later on we're going to be diving into, let's say, the state-of-the-art works, some of them from our group, where we explain how we tackle certain problems in genome analysis, and also the methods that we propose to tackle these problems. If you are interested in learning more about the analysis of biological data, these are really good textbooks, so you can check these. I can also recommend you certain papers, so you can find the links to these papers again on the in these slides, which are linked from our course page. So you can take a look at these papers to learn more about how we design, let's say, hardware and software together, or how we design the algorithm itself to achieve accurate analysis, fast and accurate analysis. So here, this is an invited paper that was published last year, where we cover essentially the current challenges for accelerating genome analysis. And we also organize workshops, invite related people in this direction, where they give certain talks and then try to motivate, let's say, why we really need fast and accurate genome analysis. So last year we've had this workshop that was organized together with Recomp, which is a leading conference in computational biology and bioinformatics field. The talks were live streamed, so you can go and watch these talks if you are really interested in that. And we of course give talks at industry in essentially the institutes. Also, we give lectures in this direction. So if you want to learn more about these directions, you can check these lectures or talks using these YouTube links. Again, many lectures, many talks. There are many resources to learn from if you are really interested in this topic. So please go and check these links and watch them if you're interested. So with that, I'll finish the first lecture today. Hopefully, you were able to essentially see what is expected from this course, what we're going to be offering in this course, and then what we're going to be covering in this course as well, and what you will hopefully improve as a student throughout this semester. With that, again, I'll finish the lecture today. We will contact with students soon regarding the project introductions, so that you can rank these projects based on your interest. And after that, essentially your mentors will contact you to make the further steps. I'm quickly checking the YouTube chat if there are any questions. There are no questions. I see some comments and hello, I guess, messages. Hello back to them as well. So I don't think there will be any other questions. So with that, I guess I can finish the lecture. So thanks everyone. I'll see you in the next weeks, either with further live streams or by premiering the lectures that we've given in the previous weeks. Okay then.